Fantastic. Thank you so much, Peter. Thanks for everyone for being here. So as Peter mentioned, my lab studied neuroimmune interactions. So basically, we look at how pain-transmitting neurons interact with immune cells. And we do that in various contexts, from allergy to cancer. And today, we're going to focus on cancer. Most of you are familiar with immune checkpoint receptor, but just a quick background. Normally, when you want to drive or start host defense, you have antigen-presenting cell that will provide co-stimulatory molecule to T cell and will eventually lead to the activation to those T cells. Those T cells will then produce responses that are specific, for example, against viruses or bacteria or cancer. The host has actually evolved means to stop the activity of those T cells to prevent autoimmune responses. So the same APCs will then provide inhibitory signals to those T cells. So basically, they will express immune checkpoint ligands that activates immune checkpoint receptor on T cells, and that will drive T cell exhaustion. So what is T cell exhaustion? The T cell will eventually co-express a lot of those immune checkpoint receptor. Classic one would be PD-1, LAG-3, TIM-3, and CTLA-4. Then the T cells will lose the capacity to proliferate, and eventually they will lose the capacity to produce cytotoxic molecules. So basically, you will have your T cells that are present but are not active. So they are not able to interact with the danger, whether it's, again, cancer, viruses, or bacteria. What immunologists and oncologists discover is that tumor cell can take advantage of these mechanisms because the tumor cell, upon the pressure of the immune system, are able to express the same immune checkpoint ligands that are then able to activate immune checkpoint receptor, the T cell, and again drive their exhaustion. And again, what immunologists and oncologists discover is that if you use antibodies that are specific for those receptor or ligand, you're able to prevent T cell exhaustion. So you drive basically, you maintain or you sustain the activity of the T cells that are then able to fight the cancer cell. And that really led to the decrease of tumor growth in various contexts. Some of the most striking data are probably done in the context of melanoma. So when you combine two of those checkpoint ligand receptor blockers, you're really able to transform responses of the immune system against the cancer. So back in the day, patients were undergo, of course, resection, radiotherapy, and chemo, but responses were actually extremely bad for those patients. So you would have about 90% of the patient that would die within 2-3 years. But now with checkpoint blockers, the patient are able to survive. About 50% of the patient are able to survive over 5 years. So I briefly introduced the immune system. So not only the immune system partake in host defense, but you also have the nervous system. And actually, when you step back and you look at both systems, you realize that they function in a quite similar manner. They detect danger, integrate and process the information. They will eventually generate a memory of these insults. And this will drive effector responses that can be either local and systemic. And these responses actually target the resolution of the inflammation. And the whole point of both of the system is actually to maintain homeostasis. So again, if you step back, you will realize that both the immune and nervous system have their pros and their cons. So for example, the immune system is highly mobile and highly specialized. However, it has a slow response time, while the nervous system allows for a very quick transfer of the information. But of course, the nervous system is actually not able to move. So from this, again, you realize that both systems are actually highly complementary. So a little bit of background on the nervous system. So we study, as I mentioned, pain-transmitting neurons. So we will call them nociceptor neuron. These are a subtype of neuron that are specialized in detecting noxious stimuli. They do that by expressing specific ion channel receptor on their surface. The most famous one would be TRPV1, which detects noxious heat. So it's an ion channel receptor. Once it's activated, it actually opens. It will allow the inflows of calcium and sodium, and that will generate action potential. So these action potential will travel from the periphery through the cell body, which are in the dorsal root ganglia, all the way to the spinal cord. In the spinal cord, these neurons need to synapse with other neurons to eventually send this information to the brain. So at this synapse, we call that electrochemical transmission, meaning that the neuron can only communicate with second-order neuron by releasing neuropeptide and neurotransmitter. Most famous one would perhaps be glutamate, but there's also neuropeptides such as substance P and CGRP. Another thing I'd like to point out is that these nociceptor neurons are classically known to detect, as I mentioned, noxious stimuli, so noxious heat, cold, and mechanical pressure. But there's more and more data that shows that these neurons can detect much more than this. They express various damage associated, they express receptor for damage associated molecules. So, for example, toll-like receptor and PAR and PRR and FPR, meaning that these neurons can also detect bacteria, viruses, and parasites. So sensory neuron, yes, they detect noxious pain, but they can also detect many other drivers, for example, bacteria and viruses. So as I mentioned, right, electrochemical transmission, when the action potential are transmitted, we need the rise of peptide and transmitter. Something that's really interesting about sensory neuron is that they are two-way road. So while they send action potential from the periphery to the brain, they also send action potential back to the periphery where the initial sensing has actually occurred. And when these action potential travel back to the periphery, they will lead to the local release of the same peptide and transmitter in the periphery. And what happened is when those peptides, think about this as the skin, for example, when those peptides and transmitter are locally released, they will act on capillaries, they will increase blood flow, they will increase vasodilatation of those blood vessels, they can drive the chemotaxis and adhesion of immune cells, and eventually they can loosen the tight junction of those capillaries. So this will lead to the extravasation of immune cells, and that will really put in very close vicinity immune cells and sensory neuron terminals. And this allows both systems to actually locally crosstalk. So this local crosstalk between the nerve and the immune cells is really what we're interested in studying, again, from allergy all the way to cancer. So neuroimmune interaction has been shown to be important in controlling a lot of different responses in host defense. And these data has been shown to be important at various barrier sites, including the skin, the lung, and the gut. So classic barrier sites that you can think of, neuroimmune interaction are really important to control those immune responses. So why we decided to be interested in tumor is we know for a very, very long time that tumor are innervated. Here is a JXMed picture from 1897 that shows the presence of nerve in sarcoma. But for over 100 years, people did not really look what would be the role of these neurons until the emergence of a new field of study, which is called cancer neuroscience. And so people have postulated that sensory neuron and neuron in general could have four main roles. Maybe they directly crosstalk with cancer cell to drive the growth of the cancer cell directly. Perhaps they control neoangiogenesis of the tumor, or perhaps they interact with immune cells and actually modulate the function of these immune cells against the tumor. And this is, again, as you can presume, this is this local crosstalk that we're interested in studying. So I'm missing a slide here. Okay, so we decided to study this in the context of melanoma. You guys are probably familiar with melanoma. It's a very aggressive type of skin cancer. And what we do in the lab is very simple. Normally, we use orthotopic cell line, which we inject in either intradermally or sub-Q. And we do that in various mouse models in which we ablate nociceptor neuron or in which nociceptor neuron are intact. Typically, we let the tumor grow. We look at the presence of itch or pain as a surrogate for the activation of these sensory neurons. And eventually, after 15 to 20 days, we harvest the tumor. We use immunofluorescence to see if the tumor is innervated. We also mash the tumor, and we do flow cytometry to look at the immune cells that represent the tumor and whether the immune function is different. So our first question is, again, whether cancer is actually innervated by these nociceptor neurons. So in this case, we use a B16F10 EGFP tumor, in which we injected in a nociceptor reporter mice. In this case, is a NAV1.8 Cree TD tomato mice. And what you can see, and this is after 22 days in the hind paw of the mice, of course, you have tumor growth. You have a lot of neuron at the surface of the skin, but you also have sensory neurons that are actually coursing inside of the tumor. So because we have these neurons inside the tumor, we ask ourselves, does a cancer cell actually change the function of these neurons? So to answer this question, we did a co-culture between sensory neuron and cancer cell. And then after 2 days in co-culture, we use flow cytometry to FACS purify each cell type, and we looked at the expression of various markers using RNA sequencing. And what we found is when the neurons are co-cultured with cancer cell, they overexpress a lot of different receptor and transmitter, including a gene that's very important for us, which is CALK. It's a gene that encodes for the neuropeptide CGRP. So we found the presence of nerve in the tumor. The tumor is able to change the transcriptome of the nerve. Then we ask ourselves, can the tumor actually overactivate these neurons? So to look at this question, we decided to do calcium microscopy. That's a technique that allows us to measure whether a sensory neuron are more or less active. Again, we use co-culture. And in this case, we load a calcium responsive dye inside of our co-culture so we can see calcium flux at the single cell level in our cells. And then we're able to flow different substances on the surface of these neurons, this co-culture, sorry. In this case, we flow classic activator of nociceptor neuron. You can focus on capsaicin, which is the agonist for TRPV1. You can see that cancer cells do not respond to capsaicin, which is expected. You can see that neuron in this case show a very little response to capsaicin because we use very low dose of capsaicin. However, when the neuron were co-cultured with cancer cell, we see very strong activation of these neurons. So this was done in vitro. What would happen in vivo? In this case, we injected our cancer cell in the hindpaw of our mice. We let the tumor grow for 2 weeks. Because we inject in the hindpaw, we're able to track these neurons because we know that the hindpaw is innervated by L3 to L5 DRGs. So we harvest specifically these DRGs, put them in culture, and again, we did calcium microscopy. In comparison to mice that receive keratinocyte, which are normal, non-tumorigenic cells, the mice that were injected with a cancer cell, we show that the neuron actually, again, present increased responses to ATP and capsaicin. So these neurons, both in vitro and in vivo, the neurons are actually hypersensitive when they're exposed to cancer cells. A classic function of the neuron is their release of peptide. I told you this is very, very important for neurons to be able to communicate with one another or communicate with immune cells. So we ask ourselves, does the cancer cell actually drive peptide release from these neurons? So again, using our co-culture, we looked at neuropeptide release. Classic neuropeptides such as VIP, substance P, and CGRP. The cancer cell alone, again, do not release neuropeptide. When you stimulate the neuron with a positive control, KCL, you see a lot of secretion of CGRP. And when the neuron will co-culture with the cancer cell, you would see that they actively release CGRP. So far what we found is that the tumor are innervated, the tumor sensitized the neuron, they will show increased calcium flux, and they will show neuropeptide release. As I mentioned, neuroimmunity is driven by peptide. So we ask ourselves, can these neuropeptides directly act on T cell and change the function of those T cells? So we look specifically at the expression of immune checkpoint receptor expression on those T cells. So we did this experiment in a lot of different ways. I'm not going to go through all these different ways. I think this is the easiest way. So we actually harvest naive CD8 T cell from lymph node and spleen, put them in culture, we made them cytotoxic, and then we expose them to CGRP for 96 hours. And then we looked at the expression of our immune checkpoint receptor on them. And we found that the T cells that were exposed to CGRP actually seems to be exhausted because they overexpress PD-1, LAG3, and TIM3, and they lose the expression of interferon gamma. And when we repeated this experiment using RAMP1 knockout CD8 T cell, RAMP1 being the receptor for CGRP, we see no effect. So again, this is flow cytometry. We think the T cells are exhausted, but to really look at whether the T cells are exhausted or not, we need to look at their capacity to eliminate cancer cells. So in this case, we co-culture B16F10 ova with OT1 CD8 T cell. I guess you guys are familiar with OT1. They have a TCR that's engineered to actually specifically recognize ova-expressing cell. So in this case, in our co-culture between B16F10 and CD8 T cell, we see that about 20% of the cancer cells are being killed when they're co-culture for 24 hours. However, if we put CGRP on the co-culture, we see that only about 10% of the cancer cells are being eliminated. So from this, we're very confident that CGRP drives the exhaustion of the T cell, right? They're less able to kill cancer cells, and they overexpress PD-1 like 3 and they lose the expression of interferon gamma. So most of this was done in vitro. Is this even relevant in vivo? I'm going to skip this, it's a little bit complicated. So is this relevant in vivo? So to do this or to ask this question, we give our cancer cell to mice that were engineered. In this case, we have mice that were engineered to not have sensory neurons. So in this case, we use a TRPV1 Cree diphtheria toxin mice. So these mice are normal, they grow normally, they can reproduce, etc. But the only thing is they do not have peptidergic sensory neuron. Peptidergic means that they are able to release CGRP. But their motor function is fine and everything. So in this case, our mice that do not have sensory neuron, we see that the tumor cell grows less and that the mice actually survive longer. And when we immunophenotype these tumor, we see that there's more CD8 T cell per milligram of tumor, which is not on this slide. But we also found that there's higher expression of interferon gamma by those CD8 T cell, and what would seem lower exhaustion, so lower PD-1 like 3 expressing CD8 T cells. So all of this points out that maybe the nerve are able to control immune responses against the cancer. So to answer this question, we repeated this experiment using two other cell lines. A melanoma cell line that's highly immunogenic, so it's a Yummer 1.7, or another cell line, another melanoma cell line, which is non-immunogenic, which is Yum 1.7. And again, in our immunogenic cell line, when we ablate nociceptor neuron, we see smaller tumor growth. But in a non-immunogenic cell line, we see no effect of the ablation of nociceptor neuron. Again, reinforcing the idea that nerve control immune responses against the tumor. So immune responses is very vast. Which immune cells would be driving these effects? So we thought about T cells. So to test this, we actually use two different approaches. So we either use an antibody to deplete CD3 T cell or deplete CD8 T cells. So bear with me here. Here's our neuron intact mice in black. Here's our neuron ablated mice in green, so smaller tumor growth as we expected. And when we eliminate T cell in both cases, we see significant tumor growth, meaning that the elimination of the neuron has no effect if we remove CD3 positive T cell. And we replicated this data if we eliminate CD8 positive T cells, reinforcing the idea that neuron would control T cell function against the tumor. So we test this in another way. In this case, we use a RAG1 mice that have no B and no T cell. And we let in, no B and no T cell. In one group, we actually eliminate nociceptor neuron. So we chemoablate nociceptor neuron using a drug which is called RTX. So what we did is we give the mice a tumor, we let the tumor grow to about 500 millimeter cubes, so a pretty big tumor, and then we transplanted naive OT1 CD8 T cells. So these were B16F10 OVA injected. And then we looked at tumor growth in these mice. And you would see that in the mice that do not have sensory neuron, these naive OT1 CD8 T cells seems again to be better able to eliminate the tumor than in mice that have intact nociceptor neuron. And again, if you immunophenotype those T cells, there's less exhaustion in those T cells. So far we did loss of function approaches. So we eliminate nociceptor neuron. What would happen if we do a gain of function, if we overactivate nociceptor neuron? So we could do this with two different techniques, either chemogenetics or optogenetics. And we decided to use optogenetics. So what we did is we use a NaV1.8 Cree channel rhodopsin mice. Channel rhodopsin is basically a protein that's derived by algae that's sensitive to a very specific laser wavelength, which is actually 472 nanometers. So the idea behind this is you can use a laser to specifically activate these channel rhodopsin expressing cells. So what we did is we activate those neurons only for 20 minutes every day, and we specifically shine the light in and around on or around the tumor. And what we found is, in comparison to naive mice that were not exposed to the laser, the mice that were exposed to laser, we see smaller tumor growth. So when you overactivate the nerve, there's bigger tumor. And this bigger tumor is actually coupled to what? It's coupled to increased CGRP content in the skin, in the explant around the tumor. So we think that we're overactivating the nerve, we secrete more CGRP. CGRP would act on the T cell, likely lead to the exhaustion of those T cells. And that's why we see a bigger tumor in these cases. So again, we use genetics. There's, of course, caveat with genetics. So can we use pharmacology to play with the system? So we decided to use a drug, which is called QX314. QX314 is basically a charged lidocaine derivative. So lidocaine is a local anesthetic. It's highly lipophilic. And what happened with lidocaine is that because it's so lipophilic, it can diffuse through the lipid bilayer of any neurons. And once it diffused to this lipid bilayer, it's going to go bind and block voltage-gated sodium channel. We all experience that when we go to the dentist. You're injected with Novocaine or benzocaine. And then, of course, it blocks your pain, but it also blocks your motor function. So it's not specific for sensory neuron. It blocks all types of neurons. So this is not ideal for us. That's why people have actually developed a drug which is called QX214 because it's a positively charged lidocaine derivative. In this case, the positive charge prevents this diffusion to the lipid bilayer. However, data have shown that when sensory neurons are actually activated, whether it's with capsaicin or whether it's with inflammation, such as the one found in tumor, those TRP channels actually open. And if you remember from the introduction, I mentioned TRP channels are actually non-selective ionic channel, meaning they will let the entry of sodium, calcium, but other positively charged molecule. Since QX is very small, it used TRP channel to actually enter these inflammation-activated neurons, bind and block voltage-gated sodium channel. So meaning we can use this drug to specifically block inflammation-activated pain neurons. So what we did is we give the tumor to our mice and we use two regimens, either a therapeutic regimen or a prophylactic regimen. Prophylactic meaning that we started the treatment 1 day after a tumor injection, and therapeutic, it means that we started the treatment, I think it was 200 millimeter cube tumors. Yep. And what we found is in both cases, when you block sensory neuron locally, so basically we injected only around the tumor, we decrease tumor growth and we also decrease exhaustion of the T cells. So PD-1 like 3 is lower and interferon gamma is slightly higher. Interferon gamma data is not absolutely fantastic, but it tends to show that those T cells seems to be more activated. So far, I hinted that we think that the effect of the neuron are driven by CGRP. So can we specifically block the CGRP receptor RAMP1 using a drug? The answer is yes, we can block this using BIBN 4096. So first of all, what we did is we did a rescue experiment. I told you that when we ablate nociceptor neuron, we ablate specifically those peptidergic neuron. So meaning that...
Sorry, I don't understand.
Seems appropriate. So we eliminate peptidergic neuron. The peptidergic neuron are the one that produce CGRP. So what we did in this case is we rescue CGRP level in the tumor once a day. So we went ahead and injected CGRP around the tumor once per day. And in this case, we're able to rescue tumor growth, not completely, but closer to what we see in intact mice. And we also see that they have normal exhaustion. So we think that CGRP is one of the major drivers of this T cell exhaustion. So what we did is we went ahead and blocked the CGRP receptor using, as I mentioned, BIBN 4096. Again, therapeutics regimen or prophylactic regimen. And in both cases, we see smaller tumor growth, less exhaustion, and more interferon gamma positive CD8 T cells. So all of this brings us to this experiment, really to nail down CGRP RAMP1 being the major driver of the effect we see. So in this case, to prove this, again, we turn ourselves to our RAG1 knockout animal that have no B or no T cell. And what we did is we went ahead and transplanted either RAMP1 wild-type CD8 T cell or RAMP1 knockout CD8 T cells. Or in some cases, we co-transplanted equal amount of both RAMP1 wild-type and RAMP1 knockout CD8 T cells. So in comparison to the mice that received the normal T cells, we see that the mice that received T cells that are knockout for RAMP1, we see smaller tumor growth. And what was really cool is that in the experiment where we co-transplanted both the wild type and the knockout T cells, we're able to track each subtype of T cell using CD45.1 and CD45.2. So within the same tumor, we're able to find that in the RAMP1 knockout CD8 T cell, those T cells were less exhausted. And to really confirm that, we actually fax purified each type of T cell and run RNA sequencing. And we can find that the ramp-1 knockout CD8 T cell, again, has lower expression of TOX and EOMs and other marker of exhaustion. So we're pretty confident that CD8 via ramp1 drive the exhaustion of those T cells. So is this relevant only in the mouse, or it's relevant even for patient? So we turn ourselves to TCGA data, and we found that melanoma patient that express high level of ramp1 seems to survive less. And then we turn ourselves to single cell RNA sequencing from patients. So at the time we carried out this experiment, 2 data sets from Aviv Regev were published using melanoma patient and analyzed by single cell RNA-seq. And using single cell RNA-seq, we can parse T cell in different populations. So we parse the T cell that were inside the tumor in either ramp1 high or ramp1 low T cells. And we found that the ramp1 high T cell in patients seems to be more exhausted. So they were expressed PD-1, TIM3, LAG3, CTLA4, etc. So it seems that in patient also the expression of ramp1 seems to be bad for T cell function. So as I mentioned, there was 2 data sets. This is the second data set and similar idea. So again, using single cell RNA-seq, we ask ourselves, is ramp1 a predictor for T cell exhaustion? We think it was the case. So we had patients that were either immune checkpoint responsive in blue and immune checkpoint resistant in red. So we could parse those populations in 2, either they responded well to checkpoint blocker or not. And then we look at the expression of various markers on those T cells. I think it was 32 different markers, classic markers of exhaustion. And when we did the delta in expression between resistant to responsive, we found that the delta in expression of ramp1 ranked actually 3rd, meaning that out of those 32 markers, ramp1 overexpression in immune checkpoint resistant patient T cell was actually quite one of the highest ones. So we think again that ramp1 might be a predictor for bad responsiveness to checkpoint blocker in patients. So based on this, we went back in the mouse to ask ourselves, if we eliminate nociceptor neuron, are we going to control immune checkpoint responsiveness using those treatments? And so basically what we did is we used our nociceptor intact inhibited mice and we treated them with anti-PD-L1. So again, our green mice do not have nociceptor. They have smaller tumor that are intact mice. Our, I guess, brown mice here are intact mice with anti-PD-L1. So they're smaller tumor growth than the intact mice. So the PD-L1 is working. But when we give anti-PD-L1 to our nociceptor ablated mice, we see much smaller tumor growth. So in this experiment, there's a little bit of caveat because we know that nociceptor ablated mice the tumor grows slower than our intact mice. So here we started PD-L1 treatment at specific time point, but we did not have exactly the same tumor size. So to address this, we repeated this experiment using, again, intact and ablated mice, but we injected our nociceptor ablated mice 3 to 4 days earlier than the intact mice. We let the tumor grow and select exactly similar tumor size, and then started the anti-PD-L1 treatment. And again, in the ablated mice, anti-PD-L1 seems to be stronger than in intact animal. So we also combine or confirm or test whether we could combine anti-PD-L1 in therapeutic treatment to block nociceptor neurons, so QX314 and Botox. And in both cases, we found that if you combine anti-PD-L1 with those pharmacological means to block neuron, anti-PD-L1 efficacy was stronger. So from this, basically, we conclude that, and I did not show you that really today, but what we found is that during tumor growth, nociceptor neuron actually start innervating the tumor prior to significant T cell exhaustion. And what we found is that the cancer cells are able to activate those nociceptor neurons using a molecule which is called SLIPI. SLIPI will act on sensory neuron. The sensory neuron will release CGRP. CGRP act on the ramp1 positive T cell and drive their exhaustion. And if we block the nerve using QX314 or Botox, or if you block ramp1 using BIBN 4096, we prevent or we decrease T cell exhaustion, and T cells are better able to fight the tumor. So we see smaller tumor growth. So from this, we ask ourselves how or whether melanoma can control nociceptor neuron function. So does it actually very clearly change the transcriptome and the function of nociceptor neurons? So to do this, we decided to do single cell RNA-seq of tumor innervating neurons. So to do this, we injected in the hind paw of our mice, our tumor. And again, we could trace these neurons from the sciatic nerve that innervate the skin. So we can trace them to L3 to L5 DRG neuron. Then we use flow cytometry to purify these neurons. So I think in this case, I think it was a NAV1.8 Cree TD tomato mice. So we sorted the TD tomato positive neurons. So we're sure that these are actually sensory neurons or neurons in general. And then we harvest that and did single cell RNA-seq. And we also have mice that were naive, so they were injected with non-tumorigenic keratinocytes. And so we found many differentially expressed genes across different subtypes of neuron. But what was really cool is we found a population of about 4 to 5% of neuron that was actually not present at all in our naive mice. And this population of neuron that was not present actually overexpressed ATF3. ATF3 is a classic transcription factor that we found after nerve injury. So if you do spinal cord injury or spur nerve injury, ATF3 is overexpressed in these neurons. And ATF3 is actually absolutely key to drive neuron outgrowth after an injury. And they do that by expressing different growth cone driver, different peptide, different transmitter. So finding ATF3 positive neuron in our tumor innervating neuron was really cool. And they not only overexpress ATF3, but they overexpress a lot of other markers that are specific for nerve injury. For example, GPR151, WFDC3, CRY-BA2. So all of these are markers of nerve injury. So many markers of nerve injury found in those tumor innervating neurons. So I wanted to confirm whether ATF3 is really expressed in these neurons. So in this case, we use an ATF3 reporter mice. So it's an ATF3 RFP mice in which, again, we injected our tumor in the hind paw of the mice, traced those neurons, and by immunofluorescence, we looked at the amount of ATF3 expressing neuron. And what we found is that, yes, we confirmed that there are ATF3 expressing neuron in our melanoma injected mice, more than our intact mice. So what would be the role of ATF3 in these neurons? What happens if we flux out specifically ATF3 out of sensory neurons? So to do this, we use an NAV1.8 Cree ATF3 flux mice, and we found that, again, these mice have smaller tumor growth and smaller tumor weight. And what we did after that is we wanted basically to measure how the immune system is actually functioning in this case. So to do this, we did it two ways, using flow cytometry and using single cell RNA-seq. So when we did flow cytometry, we found that in our ATF3 conditional knockout, we found that there was higher level of interferon gamma positive CD8 T cell, which actually match our smaller tumor growth. And we also, as I mentioned, did single cell RNA-seq. So single cell RNA-seq, of course, there's a bunch of data, but what we found is that there's lower expression of LAG3 positive CD8 T cell, and that the function of tumor-associated macrophages were also different in these mice, meaning that they express higher level of IL-1 beta, CD86, and CD80. So we think that those tumor-associated macrophages would be less immunosuppressive in our conditional knockout mice. So ATF3 is a classic marker of nerve injury. If we remove this ATF3, we prevent the reprogramming of these neurons. But how does the nerve communicate with the immune cells? One of the other things that were overexpressed in these mice was actually galanin. Galanin is another neuropeptide. Of course, you have CGRP, you have substance P, but you have about 20 different neuropeptides. So galanin is one other of these neuropeptides. And we found galanin to be very drastically overexpressed in these neurons. If you look at TCGA data, high expression of galanin in melanoma seems to be bad for the patient. So what we did in this case, I wanted to see is galanin actually important for tumor growth. So what we did is again a rescue experiment. In this case, we use again our NaV1.8 wild type mice with vehicles, so here in blue. If we flux out NaV1.8, so we see smaller tumor growth. And in this mice, in our NaV1.8 if you rescue galanin, again, once daily in the tumor, it basically rescues tumor growth all the way back to our wild type mice. So we really think that ATF3 would be upstream, inducing reprogramming of the neuron, including the overexpression of galanin. And we think that galanin would be driving tumor growth. How it would do that? So in this case, we again repeated our experiment where we harvest naive CD8 T cell, put them in culture, expose them to galanin, and cytotoxic, CD8 T cell. We found that galanin again drive the exhaustion of those CD8 T cells. So higher level of PD-1 like 3 positive CD8 T cells. And then these data are not published, so we're still working on that. But actually, we think that galanin might also be able to decrease the capacity of T cell to move towards CCL19. So galanin might be able to, in one case, drive the exhaustion of the T cell, but in a second case, actually change the kinetic of those T cells, so are potentially less able to move towards chemokines. So that could be a second mechanism by which galanin actually impair immune function. So why was this actually interesting? Is because our colleague Moran Amit at MD Anderson is an head and neck surgeon, and he had a very specific population of cutaneous squam population. And those cutaneous squam patients were stage 2 to 4, and they were treated with anti-PD-1. And so basically, he had samples before, during, and after treatment. And of course, some of the patients respond to the treatment, some of the patients do not respond to the treatment. And when you harvest the patient a tumor and you do basically immunofluorescence, immunohistochemistry, and electron microscopy, we found that in a non-responsive patient, we see higher level of perineural invasion than in responsive patient. What is perineural invasion? Is basically you will have in the tumor, as I mentioned, there's nerve growing, and the tumor is actually able to engulf or really to wrap around these nerves. And that will lead to what we think, including, it will lead to Wallerian degeneration and also to nerve injury. So finding this nerve injury phenotype in the patient was really, really interesting. And actually, when Moran stained for ATF3 expression in these patients, he found there's higher ATF3 expressing beta-3 tubulin positive neurons. So again, you're able to find in the patient cohort that ATF3 is overexpressed in these patient samples. So what we did is we actually generated a signature for nerve injury based on our single cell RNA-seq. And when we looked at this nerve injury signature, we looked at different cohorts and see whether it correlated with good or bad responsiveness to anti-PD-1 in patient. So in this case, we had, as I mentioned, the head and neck patient that were that had perineural invasion, and whether they respond or not to treatment. And this nerve injury signature is enriched in patients that do not respond to PD-1. If we looked at anti-PD-1 treated patient with melanoma, again, the nerve injury signature is enriched in those patients. In a gastric cohort cancer patient that were treated with anti-PD-1, again, enriched, and another cohort of melanoma, this nerve injury signature is enriched. So what we finally did is a spatial transcriptomic in these head and neck patients. So we had our nerve injury signature. So the genes that are involved in this nerve injury, we see them in green. We had genes that are involved in or correlated with immunosuppression in red. And we had genes that were basically known to drive anti-tumor immunity in purple. And this is, I think, really, really cool, is that across samples, you see that the nerve injury signature really overlap very well with immunosuppression. And actually, the opposite is present. When you don't have this nerve injury signature, you have a very, very nice anti-tumor immunity signature in those patients. So again, I'm not showing you all the data, but what we found is that when the neuron grows in the tumor, there's perineural invasion that leads to Wallerian degeneration. It changes the transcriptome of the neuron, they will express ATF3, this nerve injury marker. ATF3 will drive the overexpression of different peptides and transmitter, including galanin. We found that galanin act on T cell and drive their exhaustion, but we also found that these neurons overexpress IL-6 and interferon 1. Again, did not show you, but we found that IL-6 and interferon 1 are able to act on CD163 positive tumor-associated macrophages and drive an immunosuppressive phenotype in these macrophages. So what was really cool is when we published a paper in August. So this was done in cutaneous squamous cell carcinoma and melanoma. But there's another group that published a completely different paper that also found that nerve injury seems to be a signature for glioblastoma progression. So it seems to be very important across different cancer types. So I'm going to show you now a little bit of data that would look at how we can use this tool that's developed by Ben HK at University of Toronto, which is called predict.io.ca, which is really, really cool because normally when we look at TCGA data, we only look at one gene, right? Is RAMP1 good or bad for patient? But what Ben HK developed is you can look at you can look at a signature. So we can look at several gene expression and whether this is good or bad for patient. So we came up with a neuron signature, not injury neuron, but neuron signature. So 8 genes that are expressed by sensory neuron. And we found that this nerve signature in cancer patients, so across cancer, this is not only melanoma, but really across cancer. So you see melanoma, urethral, you see bladder, kidney, blah, blah, blah. So across cancer, this nerve signature seems to be very, very bad for patient. It actually ranked higher than T cell exclusion, which is this dot. So really, having a nerve signature seems to be super bad for patient. And does this nerve signature actually correlate with responsiveness to treatment? It seems to be the case, where having a nerve signature, an enrichment of this nerve signature, seems to be bad for immune checkpoint receptor responsiveness in melanoma. No effect in the lung and kidney, but in melanoma seems to be bad. So I think maybe should we go with question now or? No. Yeah? All right. You guys still, yeah? Okay. You got, okay, quickly on this. So primary melanoma, we found they are innervated. We find that, the nerve are bad, but what would happen in metastatic melanoma? So in this case, we injected, actually this is work led by Alex Breber at Madison, Wisconsin. Alex, what he did is he injected melanoma IV. When you inject melanoma IV, it goes seeds pretty much everywhere, but it seeds a lot into the lung. So what he did is he looked at the presence of nerve into those metastatic lung. He found that there's higher nerve content around the tumor and in the tumor in those lung in comparison to normal lung. So there's more neuron in the tumor and around the tumor in the lung. What happens if you eliminate these nociceptors? So if you chemoablate them using RTX, you see that there's less lung metastasis. He actually replicated these data using chemogenetic. In this case, is a NaV1.8 HM4DI. So it's a mean to block sensory neurons specifically. And again, he found that there's smaller tumor growth in those lung mets. So the part that's, I think, super cool about this project is if you looked at metastatic patient, the patient that are dead, lung metastatic patients, the patient that are dead, it seems that RAMP1 is overexpressed on the tumor itself. So you see higher expression of RAMP1 on the tumor itself in those patients. And again, in metastatic melanoma, you see that RAMP1 expression is bad for the patient. But the part that's super cool is if you compare normal melanoma, if you see it in the skin, you have very, very low level of RAMP1. But if you injected IV, see it in the lung, you harvest those tumors, you see that they drastically overexpress RAMP1. Remember, RAMP1 is a receptor for CGRP. And what would happen if you eliminate RAMP1 from these tumor cells? So in this case, you CRISPR knock out RAMP1 out of the tumor, out of the melanoma. So it's a YUM1.7 RAMP1 knockout. You see that there's less lung metastasis in these mouse. Another thing that he found is that if you look at single cell data, you will see that PGFR beta positive cell actually also overexpress RAMP1. What is PGFR beta? It's a marker for pericytes. So PGFR beta positive cell have more RAMP1. But the other super cool thing is if you compare a normal mouse, you harvest the pericytes and you looked at RAMP1 expression, it's very, very low, almost nothing. But in tumor-bearing animal in the lung, RAMP1 is drastically overexpressed on those pericytes. And then he was able to show that CGRP, the neuron themselves, are in very close vicinity to those pericytes in lung metastasis. And finally, he used Cree-Lox to flux out RAMP1 out of those pericytes, and he found that there's smaller tumor growth in those pericytes. So all of this, and the paper was submitted, and we got a super difficult question from one of the reviewers. So we're quite sad about that. And they asked us to actually carry out a clinical trial using the RAMP1 blocker in metastatic melanoma, so which was kind of a crazy question, which of course we would love to be able to do, but this would take years. So, but Alex was able to access basically 147 healthcare organization, and the major one was the VA hospital. So basically the Army hospital in the US, which has tons of patients. And it turns out, so this is over 160 million patients. And turns out that some of those patients were actually having melanoma and were treated with our CGRP blocker. Why patients are treated with CGRP blocker? Because this CGRP blocker is used actually for the treatment of migraine. So it's a very good treatment for migraines. It's been accepted for 5 years. And we're able to find 119 patients out of those 160 million patients that were treated with the RAMP1 blocker. So when we compare these 120 patients to metastatic melanoma that were not treated with those drugs, so there's a lot, obviously, the CGRP blocker seems to be increasing the survival of those patients. So hopefully this will answer the reviewer's question. I hope so. Anyway, so all that to say, and I'm going to conclude on this. Again, this is led by Alex. He found that during metastatic melanoma, the cancer cell actually themselves overexpress RAMP1. He found that nociceptor neuron would secrete CGRP. Did not show you those data, but CGRP will act on the cancer cell themselves, drive the proliferation and migration of the cancer cell in the lung tumor. And he also found that pericyte in the lung overexpress RAMP1. They are able to receive signal from CGRP positive neuron that will drive proliferation and angiogenesis of those pericytes. So we think that also contribute to tumor growth in this case. And on this, I'm going to conclude by thanking people that did the work, Mo and Mariam that led the first project, Tuani that led the second project, and other people in the lab right now, which we have Anais, and we have Serbi, and we have Alyssa, and Isaac. Are you on the picture? All right. And we should Photoshop you, buddy. All right. So thank you, guys. Anciano. Oh, Anciano.
You don't remember anything else from this talk? 1 in a million. Go buy your lottery tickets. That's amazing.
Yeah.
So please go ahead. Anybody has a question?
Thanks for a great talk. I'm just wondering, is the propensity to see that sort of neuronal associated mechanism dependent on if sort of primary tumors are innervated? Like, are there sites where there's less innervation and you don't see that same effect?
Yeah, no, that's a great question. There's definitely tumor that have a lower neuron injury signature or neuron signature themselves. So when you looked at every type of cancer using TCGA data, I don't remember which one were lower, but I know for a fact that what I can tell you, for example, is we did preliminary data using leukemia, and we see the opposite effect in leukemia, where when we block nociceptor neuron, we see actually a smaller tumor growth. And there's definitely tumor that are more painful and tumor that are less painful, right? You can think of osteosarcoma being very painful and pancreatic cancer or ovarian not being very painful, right? So that said, we do see sensory neuron in ovarian cancer, but yeah, it probably depends by cancer and by location, and not to overlabor that, but nociceptor neuron there's three main origin from these neurons, and the innervate their transcriptome is very different. So the one that innervate your internal organ are different than the one that innervate your skin and your face, basically. So they have different transcriptome and function. So you would think that they would have different function on the immune system too.
I should go Marco first, trainings first.
Thanks. Just as you were saying, like cancer is an incredibly painful disease. Pain is treated almost in every type of patient through whether that's something simple like Advil or something more complex like opioids or something.
For sure.
Is
Yeah.
there a lot that's known about how those more routine pain management strategies could influence?
So yeah, so we have with a colleague, we have a JITC paper on opioid and cancer responses, if you want. It seems to be very bad for two reasons. And the main reason is that many of your immune cells actually express opioid receptor. And it seems that those opioid drugs would directly act on your immune cells and dysregulate their function. So it seems that opioid themselves, independent of the nerve, they're probably bad for immune responses. But of course, a lot of the patient, it's routine. After surgery, 99% of your patient will get opioid after the resection of the tumor itself. So this is something that that needs to be looked at if we can use other pain treatment. And if you think about Advil, it's not really it's an anti-inflammatory, right? It's not really a pain blocker itself. Now there's a new NaV1.8 drug that is able to block sensory neuron itself. It's been released last year by Vertex. I don't think someone has looked into that yet, but that's a cool idea that if we target specifically the neuron, maybe that will be better both for the immune responses and prevent this negative neuroimmune interaction. Yeah.
Great talk. So in your immune checkpoint response analysis across different cancers, you showed that kidney cancer is distinct compared to melanoma and other cancers.
Yeah.
And it's known that kidney cancers are highly infiltrated, but they respond poorly to immune checkpoint compared to melanoma, which is again highly infiltrated and responds well. What's going on in those two things in terms of your?
Yeah, no, I don't think I should not say that. I know there's someone working on kidney cancer, but it's not out yet. But yeah, it's a good question, I guess. Like no one has really I guess bouncing back on the previous question, is it different types of innervation, or there's nothing to do with innervation, or different timing of this innervation? So this was a little bit surprising, though, the data you're referring to, where when we see no effect of the checkpoint blocker, because if you look at TCGA data specifically for CGRP and substance P indiced peptide, I remember that kidney cancer was one of the highest one where we thought we would see effect, meaning like high expression of those peptides seems to be very, very bad for the patient. So this was a little bit surprising to find this metadata actually. So, but I think someone needs to test it in the mice, really. No.
Is there any link with lymphovascular invasion at all?
Yeah, great question. No one has looked into that in this context. Yeah, that's another thing that's very, very relevant.
Very next talk.
Thank you, Major.
My question here, in your early studies, but the animal studies, you looked at tumor volume. Did you do a long-term looking at tumors? And the reason I'm asking that is because a lot of the neuropeptides induces cancer cell become metastatic. And I'm just wondering if you looked at that in the long term or not.
The longest we've done was 21 days because it's just reached IACUC guideline. So those B16F10 and whatnot, they grow so fast. So it's probably not the best model, to be honest. I think you're right. I think slower growing tumor would be much more relevant for us for many reasons. For example, the nerve probably grow very slowly inside of the tumor as well, right? So having like a genetic model where you have a slow growing tumor would make much more sense. I think you're right. something we need to do. We bought those. It's a triple combination tier, ERT, B16, yeah. But we had those mice for years, and no student has ever tested them. So yeah, so we should, yeah. But your point is, yeah, it's a good point.
The other question I have is that, the neuron immune cell cross-on,
Yeah,
who's in control? Is the immune cell?
that's great. Yeah,
Triggering the.
It's a great question.
neurons to put out peptide?
great question. Great question. Yeah, we know that. So Myron is pointing to a fact that neuron express a lot of cytokine receptor, and we know that they sense a lot of signal from the immune cells themselves. So is it the cancer that drives neuron activation and then on the cancer, or is it the immune cell that acts on the neuron? That's a great point, and I don't think we don't know, and I don't know how we could test this directly, right? Yeah, it's a tough thing to test because the cancer cell can activate neuron in so many ways. Either growth factor, we found sleepy, we also found FGF1 that could activate neuron. So there's so many factors that are secreted by cancer cell. It would be hard to block all those signals and see, do we see the same thing with the immune cell only active on the neuron? But yeah, that's a good question for sure.
There would be wonderful ways of actually getting at these connections you've described to us. And early on, you talked about a biomarker, a SLIPI, I think.
Yeah.
Comes from tumors and activates nociceptor neurons.
Yep.
So can you look at a biomarker like that quantification to see whether a particular tumor might engage this type of mechanism more than others?
Yeah, that's a great point. I think that's another thing that no one in the cancer neuro field has really looked into, but I think the Human Protein Atlas has a lot of these biomarkers that we could look at whether this correlate with activation of the or involvement of the neuron in those cancer. I think it's a good point. That said, peptides are super peptide themselves. For example, CGRP will not be a good target because they're metabolized very quickly. So maybe a sleepy would be a better biomarker, but I think it's a very valid point. Yeah.
So the jury is left, so we can ask you about tertiary lymphoid structures. Now, you did mention.
Yeah.
Slide, but I'm surprised you didn't pester you about that.
So there's a separate group that actually, what's his name? Yuri Blemovich at University of Pittsburgh that actually ablated nociceptor neuron in melanoma and found less TLS in those melanoma. And he also found that in patients that have high innervation, they have less TLS as well. So both in the mice and the human neurons. Yeah.
CNS question.
Sure.
So do you have these neurons in your brain?
Yeah.
No,
well, not sensory neuron themselves, but you do have, well, obviously you do have neuron in your brain.
Nociceptor neuron.
Not in a brain matter you have in your meninge, right? If you have meningitis, this pain, so
something like glioblastoma, you would not expect there to be engagement of this type of mechanism. Yeah.
Well, that's a project we actually have in the lab where maybe not directly in the brain, but you could think that these neurons would be engaged somehow and control immune responses in the periphery, and that will impact immune influx to the brain. And I know there's 2 different papers that has been, not by us, unfortunately, has been submitted with neuron ablation and glioblastoma, and they see pretty striking effect actually, where this peripheral crosstalk would be important for glioblastoma progression. Yeah.
Well, from that cohort from the VA, it might be.
Right.
Able to find
Yeah.
Any tumor.
Yeah. Absolutely. Yeah, that's true.
One last question, especially with clinical trials with patients. One of the hot areas that's coming up now is the platelet-derived thromboxane. They have shown that thromboxane induces cytotoxic T-cell immunosuppressant.
Okay.
Shuts it down.
Uh-huh.
Maybe this could be another area that when you're doing patient studies.
Yeah. Yeah. Okay. Yeah. Well, thank you.
Not seeing any more hands, so please join me in thanking Sébastien.